afternoon, everyone. How are you?
Good afternoon,
Good
ma'am.
afternoon.
Good afternoon. How are you all? How have you been?
We are
Fine,
fine.
ma'am. What about you?
I'm also good. So today I have something for you, a very beautiful picture which says a lot about itself. So just look at the picture and tell me, what do you infer when you see this picture? What thoughts come to your mind?
Family.
Diversity.
Yeah, one by one, please. What thoughts come into your mind when you look at this picture?
Family.
Family, very good.
Diversity.
Diversity, very good point. Anything else?
Ma'am, variation we have among us.
Variation, very good. Variation among each other or variation among one individual to other when we see them. So these are the points which come into our mind when we look at this picture, that we all look so different that we are diverse. The biological world, or we as humans, are so diverse that each individual is different except for the identical twins. And when we talk about variation, the diversity is caused by variation. And family, how do you infer, what do you infer about family when you look at this picture? What about family? If we look, if we consider variation, do you find variation within a family, or the family is alike?
We find that
And there are variations.
There are variation even within a family. So let us see what is the basis of these variation and what is this that causes these variation. So we are going to dwell deep into the molecular basis of this inheritance or these variation, that which molecule is basically going creates these type of variation or this much of diversity in this kingdom. You would be surprised to know that you, me, or your friend, that means a human is 99.9% similar to another human, and still we are unique. Which percentage is left? If I consider that 99% of the genetic similarity lies between two individuals. So how much differences are there? What percentage?
0.1%.
0.1.
0.1%. So is it large or is it very small fraction?
Very
Small.
small.
Very
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 small.
small fraction, but you can appreciate the beauty of nature or the beauty of molecular biology. Thus only this 0.1% of the differences is accountable for the variation which we see around. So in humans, 99% of the 9 genes are similar to the person sitting next to us. That means I am 99.9% similar to you, and you are 99.9% similar to your friend sitting next to you. The rest, that is this 0.1% of genes, this tells us everything from our eye color to whether we will be having any disease or what will be our skin color, what would be our behavior. So everything which makes us unique is decided by 1/10 of the 1% differences in DNA, that is 0.1% of the DNA, which is accountable for making all of us unique. Isn't it amazing? A biomolecule or a macromolecule, but the differences at the micro level, micro differences in a macromolecule like DNA is responsible for the differences which we see around, right? You are familiar with this picture. Can you just name this person? Who is this? A famous personality on a web series.
Dr. Salunke.
Dr.
CID.
Salunke, very good, Dr. Salunke. And what was the occupation or profession of Dr. Salunke? What was he doing in the series as an actor?
Forensic scientist.
Forensic scientist.
Very good. He was a forensic scientist. So can anybody tell with which raw material the forensic scientist, or which is the biomolecule which is of interest to a forensic scientist? क्या check करते हैं ये लोग?
DNA.
Yes,
DNA.
exactly. They check the DNA, that is deoxyribonucleic acid. And this is the basis for all variation. That's why they check it, because they need to identify the criminal or the suspect, anybody. So we can, on a molecular level, differentiate one individual from another on the basis of DNA. So today we are going to do the topic that is molecular basis of inheritance. So what according to you is the basis of inheritance?
DNA.
DNA.
Very
DNA.
good. It is DNA. So in this molecular basis of inheritance, we involve the study of genes, DNA, DNA as a genetic material, the DNA replication, its transcription, its translation, genetic code, regulation of gene expression, and many more topics. So we are starting the exciting topic, that is the structure of DNA. And today's class would be a little bit different. I am not going to teach you anything. Rather, we will explore in a collaborative inquiry manner that how a DNA looks like, or what is the chemical composition of DNA. So I am not going to teach you anything, but we will together explore the structure of DNA in our today's class. So I'm sure that you want to learn more by this time, because the interest is quite high that what is DNA. So let's, without further ado, we should see what is DNA, where does it resides. So what is this? What is this? This is a cut open
Cell.
cell. Very good. This is a cut open cell. When we open a cell, in the center of an animal cell, what do we find?
Nucleus.
Very good. We find nucleus. And when we delve deeper into the nucleus, inside the nucleus, genetic material is found in the form of
Chromosome.
chromosomes. And the chromosomes are composed of a chemical biomolecule, chemical structure. Chemically, it is composed of a biomolecule, and that biomolecule is called
DNA.
DNA. So this would be our further investigation, that what is the chemical structure of DNA. So are you ready?
Yes,
Yes,
ma'am.
ma'am.
Yes, ma'am.
Because today this is up to you, that we are going to discuss what is the structure of DNA. Nobody is going to teach you. We will explore. So let's see. And this method is called process oriented guided inquiry learning, or POGIL. So we are going to explore the structure of DNA. Just take a intense look at the model 1, that is structure of DNA. Look at the diagram and the components very carefully. Take half a minute to explore the diagram in detail, and then we will explore few questions pertaining to this diagram that will lead us further to the structure of DNA automatically. Right? So your time starts now. Just take 30 seconds for a deep overview of the structure. Look at the diagram and the components very carefully. Yeah. So that time is up. I hope you have seen the diagram.
Yes, ma'am.
And all the components of
Yes.
diagram carefully. Now, pertaining to this diagram, we'll be exploring few questions together, and those questions will lead us further in exploring the structure of DNA. And by the end of all those questions, the entire structure would be with us, and then we would consolidate it as a group, that what is the structure of DNA. So let's start. Are you ready for the questions?
Yes, ma'am.
Yes, ma'am.
Yeah. So again on the screen, there are questions which we will be exploring further. So refer to the diagram given here in the model 1, which you just explored a bit. Delve deeper into it pertaining to the question and try to answer few questions. So first question, what are the three parts of nucleotide? So may I ask any one of you?
Yes, ma'am.
Yeah, Anupama, can I have it from you? What are the three parts of a nucleotide? Yes.
Oh, yes, ma'am.
Yeah.
The three parts of a nucleotide are phosphate, deoxyribose sugar, and nitrogen containing base.
Phosphate, deoxyribose sugar, and what
Nitrogen
else?
containing base.
Nitrogen containing base. So here we go with the first question, that a nucleotide is composed of a phosphate, deoxyribose sugar, and a nitrogen base, right? Now the next question, what kind of sugar is found in a nucleotide? Look at the picture again and tell me what kind of a sugar is found in nucleotide. Can I have it from you, Ria?
Ma'am, deoxyribose sugar.
Very good. So the sugar is deoxyribose sugar. This is a pentose sugar containing 5 carbon molecules. So this is the 5 carbon containing 5C deoxyribose sugar. This is a pentose sugar. Now the next question is, which nucleotide component contain nitrogen? Out of the 3 components, which has nitrogen? May I come to Madhavi? Can you answer this question, beta?
Ma'am, nitrogen containing bases.
Exactly, the nitrogen containing bases. So which part of nucleotide component contain nitrogen? It is the nitrogen bases. So nitrogen is found in bases. Next, what are the 4 nitrogen bases found in model 1? Yeah. Yes, Arshia, can I have it from you?
Yes, ma'am. Ma'am,
Yes.
adenine, guanine, thymine, and cytosine.
Adenine, guanine,
thymine.
cytosine, and thymine. Very good. So this brings us to the first question. These were the 4 parts. So can anybody consolidate the learning from these 4 questions? Anyone? Yeah, Ria, can you consolidate what we have done or what we have learned out of these 4 parts of question number 1?
Ma'am, we learned that the nucleotide is made up of three parts, which is phosphate, deoxyribose sugar, and nitrogen containing base.
Very good.
And the bases are of four types, namely adenine, thymine, guanine, and cytosine.
Very good. So this is the learning that the DNA is made up of nucleotides, and one nucleotide is made up of phosphate, deoxyribose sugar, and a nitrogen base. This is one nucleotide. The nucleotide contain deoxyribose sugar, and the bases contain nitrogen. The nitrogen bases are of 4 types: adenine, guanine, cytosine, and thymine. Now here comes the next set of question number 2. So look at the ladder model of the DNA and circle a single nucleotide on each side of the letter molecule of DNA. So since the rights of writing are with me, just let me know if you agree whatever I have circled. Is it correct?
Yes, ma'am.
Yes, ma'am.
So what is
Yes,
this?
ma'am.
What I have circled is a
Nucleotide.
Nucleotide.
very good. I have circled a nucleotide. So nucleotide, what is this in nucleotide?
Phosphate.
Phosphate. What is this?
Deoxyribose.
Deoxyribose sugar. And what is this?
Nitrogen containing
Nitrogen.
base.
Nitrogen containing base. Very good. So this is our what part of the nucleotide make up the rugs of ladder? Rugs means जिस पे हम चढ़ते हैं सीढ़ी पे. So what makes the rugs of ladder?
Ma'am, the nitrogen bases.
Very good. It is the nitrogen bases which forms the rug of DNA ladder. Now next question, what part of nucleotide make up the side bone of the ladder? Yes, Aushadhi, can I have it from you?
Yes, ma'am.
Tilt your camera a bit, we can't see you properly. Yes, Aushadhi.
Um, Ma'am,
Yeah, the
deoxyribose.
question was, what makes up the sides or the backbone of the ladder of DNA?
Ma'am, phosphate,
Very good. And
deoxyribose sugar.
very good. So sugar and phosphate forms the backbone, whereas nitrogen bases hangs inwards. Okay. Now look at the bottom and the top ends of a ladder. Look at it. ध्यान से देखिए. Look at the top and the bottom end of the ladder. Are the rugs parallel? And the clue is, look in the diagram, do the ends of the strand match or they are anti-parallel if their ends do not match? Look at the diagram carefully, analyze for a while and then answer. Let me come to Ria. Ria, can I have it from you?
Ma'am,
Are the ends parallel or they are anti-parallel?
I think that they are
Swati is also there.
Ma'am, they are anti-parallel because the position of phosphate is different.
Very good, because the position of phosphate is different. What is parallel? When anything just looks like this. When they overlap each other, then it is called they are parallel. But they are like this. They are not parallel. One side is not matching the other side. So they are anti-parallel. Very good. So what should I write? Both the strands of DNA ladder are
Anti-parallel.
Anti-parallel.
Very good. They are anti-parallel. So what is the consolidation of the second slide? Can anybody, Namya, can you consolidate? Can we have you on screen, beta? Switch on your video if it is off. Yeah, thank you so much, Namya. Namya, please look at the slide again and consolidate whatever we have learned in this slide. Starting from A part. What is a nucleotide?
Ma'am, the rungs of the ladder are made up of nitrogen bases and the backbone is made up of phosphate and deoxyribose sugar.
Very good.
And they are anti-parallel because the position of phosphate is different.
Okay. What are anti-parallel?
Ma'am, the ends do not contain the
What is
same
anti-parallel? It is the DNA strands are anti-parallel. One DNA is made up of two strands which are twined or which are like ladder. They may be like a helix or they may be like a ladder. So there are two strands of a DNA molecule and they are anti-parallel. So let's go back to first slide, what we have learned till now, that the DNA is made up of two chains. The chains are composed of nucleotide. Nucleotide is composed of phosphate, sugar, and nitrogen base. And these nitrogen bases are of 4 types: adenine, guanine, cytosine, and thymine. There is a trick to remember that Anju Goyal ne TC Leli. Anju Goyal is a girl who has taken TC. So Anju Goyal has taken TC. That is adenine, guanine, cytosine, and thymine. And then the backbone of the DNA molecule is made up of phosphate and sugar, whereas nitrogen base forms the rugs, or they hang inward. Yeah, and both the chains are anti-parallel. So let's proceed further. Can anybody let me know? I want to label these. What are these nitrogen bases? So I'm asking Khyati, can I have it from you? Please help me in deducing this answer. So what is this? What should I label it to as?
Nitrogen containing base.
No, just look at these 4 base pair, and particularly which nitrogen base is this?
Cytosine.
This is
Cytosine.
cytosine. Very good. And what is this base pair?
Guanine.
Guanine. Yeah, now Namya, please help me in deducing this. What is this base pair?
Ma'am, thymine.
Thymine. Very good. And what is this?
Ma'am, adenine.
Adenine. Again, going to this trend, this is A, this is T, this is G, this is C, this is C, this is G, and this is T, this is A. Am I right?
Yes, ma'am.
Yes, ma'am.
So now explore that the 2 strands of DNA are held together. So this is done, right? When one nucleotide contains adenine, what type of base is adenine attached to on the opposite strand? If on one strand it is adenine, what would be the other nucleotide contain?
Thymine.
Yes, very good. It would be thymine. So adenine pairs with thymine. Similarly, when the 2 strands of DNA are held together, they are held together with the help of hydrogen bonds, as the question says. So it is obvious that these bonds, which are gluing them together, are hydrogen bonds, and these are weak bonds between the polar molecule. How many hydrogen bonds connect the 2 bases in question number 4? That means adenine and thymine. How many nitrogen bases are there? How many hydrogen bonds are there connecting this?
2.
Very good. It is 2 hydrogen bases. Very good. Now coming to this. When one nucleotide contains cytosine, what type of base is there on the opposite strand? Look at this again.
Guanine.
Very good. It is guanine. So we can say that the cytosine pairs with guanine. How many bonds are there when we see this? How many bonds are there in between? This is cytosine, this is guanine. How many hydrogen bonds?
3.
3.
3.
So we can say this pair bonds together with the help of 3 hydrogen bonds. So now we have enough with us to consolidate the learning. So I would request a pair of 2 students to just give me few pointers on the DNA structure. So I'm making pair of two girls, like Arshia and Aushadi. You can come as a pair, and both of you come on the screen. Rest all can switch off their video for a while, so that these 2 girls appear on the screen. Yeah, thank you so much. So this is my pair. So start with the structure of DNA, which we have just learned. Consolidate what we have learned. 4 statements about the chemical structure of DNA, one by one, starting from Arshia.
Yes, ma'am. So DNA is a nucleotide is consists of a phosphate group, a deoxyribose sugar, and a nitrogenous base.
Very good. Next, Ashadi.
Ma'am, the nitrogen bases contain four type of nitrogen bases, namely adenine, guanine, cytosine, and thymine.
Very good. Very good. Next two girls can come up on the video. Next pair, please. So can we have Ria along with anybody can come up with Ria? Ria, you choose your pair. Please take one girl with you.
Ma'am, Khyati.
Yeah, Ria and Khyati, please continue with the next set of structure. So what they said, Ashadi and Arshia, they just told that the DNA is made up of two strands and each strand is made up of nucleotide. So the DNA is a polynucleotide. Each nucleotide is comprising of nitrogen base, sugar and phosphate. There are four types of nitrogen bases: adenine, guanine, cytosine and thymine. So just from there you just go on.
Ma'am, the DNA structure.
Kathy, can we have you? Or Namya can come if Kathy is having any problem of network. Namya, please come with Ria. Yeah. Yes,
Uh,
Ria.
Ma'am, the DNA structure will be made up of two chains of nucleotides,
Yeah.
and they can be parallel or anti-parallel depending whether uh, the same type of arrangement is there on both the sides.
No, it is the DNA chains are anti-parallelly arranged. Right. Now, what about the nitrogen? Ria?
Ma'am, in adenine and thymine, there will be two hydrogen bonds, and between cytosine and guanine, there will be three hydrogen bonds.
Very good. So they are bounded by 2 and 3 hydrogen bonds. Very good. So let's consolidate on a slide that what we have learned. You all can switch on your videos, please. So this is what we have learned by this inquiry together, that chains, this is one chain and this is another chain. Each chain is composed of nucleotide and a nucleotide is composed of a nitrogen base, a sugar and a phosphate. They are anti-parallel and generally the nitrogen bases are can be grouped TC. Anju Goyal has taken TC from school. So this is purines. What are purines? Look at this structure. Do you find any difference between the nitrogen bases? What is the difference between the purine and the pyrimidine? The gross difference, what you can see? Yes?
Ma'am, the purines have double ring structures, and ma'am,
Very good.
pyrimines have single ringed structure.
Pyrimidine are single ringed, whereas purines are double ringed. So when a purine, there as of rule, what you said, A combines with T and the G combines with C. So what you can say, which group is pairing with which group? A purine is combining with a pyrimidine. So this is a base pair rule that A combines with T, C combines with G, or we can say one purine with one pyrimidine. So when there is always one purine and one pyrimidine, what would be the distance between these two? Would this distance vary or this would be same all the time?
It will vary.
It would vary. Anybody else? What are your takes? One purine always combines with one pyrimidine. That is one ring of purine, two rings of pyrimidines. They combine with each other, making a three-ring distance. So what do you think? Would it vary? Means two-ringed purine and a single-ringed pyrimidine, they combine always. So there would be always a three-ring distance. So would it be constant or it would differ?
Constant.
It will be constant.
Constant. So the distance between the two DNA strands is always constant because it is always one purine combining with one pyrimidine. And as we had discussed, between A and T, how many hydrogen bonds?
Two.
And between C and G?
Three.
3. Very good. So here is the consolidation. What we have learned today will be with you forever if we just sing it along in a form of a song. So I know Arshia has a very good voice. So we all can be after Arshia, and Arshia, you can take the lead in just encapsulating what we have learned. This DNA song would keep it us in our memories for long, the structure of DNA. So this is a DNA song which belongs to all of us because this is the basis of inheritance what we received from our parents, and this is the basis what we will give to next generation. So this was all about the structure of DNA, and I hope you all have learned the structure of DNA. Now Arshia, we want to hear it in your beautiful song, beautiful melodious voice. Now you sing along so that we can seep it in our memories, please.
We love DNA made of nucleotides, sugar, phosphate, and a base bonded down both sides. Adenine and thymine make a lovely pair. Guanine without cytosine would be awfully bare. We love DNA, its life's genetic code. Instructs each cell on how to make proteins by the load. Adenine and thymine in DNA make a pair, but in RNA where there's adenine, your cell is there. We love DNA and its fine base pairs.
Thank you so much, Arshia. It was so lovely. It was so lovely. So I have composed these songs for my students only because I feel that these songs just bring it to our long-term memory rather than a short-term memory. So I hope you all have understood the structure of DNA. We would continue with the structure of RNA tomorrow, right? Thank you so much.
Thank you, ma'am.
Thank you, ma'am.
And please,
Thank you, ma'am.
assignments are
Thank you,
there.
ma'am.
Just attempt those assignments so that we can understand or we can consolidate our learning to the best possible manner. Thank you so much for joining.